The most important topic called as the neurulation. To just give you a description about this neurulation, first of all, after fertilization, it will form a single cell structure called as the zygote. This zygote will further divide and forms a compact structure called as the morula. And this morula, further forms a cavity with a group of cells in between, which is nothing but called as the blastula. And this blastula further undergoes gastrulation to form a structure called as the gastrula. So this is the outline of the gastrula, how the gastrula appears. With the intervening tissue. So within this gastrula, I will just write it as gastrula. Within this gastrula now you can see the three layers, that is nothing but the ectoderm, this is nothing but the mesoderm and the endoderm. So all the three germ layers are seen within this gastrula. From this gastrula now the neurulation occurs, and this neurulation will lead to the formation of the notochord and also the neural tube. So if you take a section at this level, how it appears we will see here. So to show you that, I'll just draw this one. This is the ectodermal part, whatever you are seeing. And now just below that you will be seeing another structure called as the mesoderm, and below that you will be seeing the endoderm. So I'll just write it as, this is nothing but the ectoderm, this region is called as the mesoderm, and this region is called as the endoderm. And also here you can see a tail-like structure is seen here, right? A tail-like structure. This is nothing but called as the caudal end, and opposite one is nothing but the cranial end. So now what happens is, like, during the development, typically a small primitive streak starts developing from the caudal end and it progresses towards the cranial end, and further it progresses, and in between it forms a depression. Please remember, it is a depression called as the primitive node. So this is nothing but the primitive streak, and this is the primitive node. Now the thing is, like, if we take a section like this, which will pass through this primitive streak and primitive node. If I have to take a section like this, if I cut a section and see from the side, how it appears is, this is a cut section of this one. So now I said that this is not an elevation, this is a depression. So I am showing you the depression here like this, and now this is all the ectoderm part, right? So this is the ectoderm. So within the ectoderm only this depression is seen, which is nothing but the primitive node. And below the ectoderm you will be seeing another layer called as the mesoderm, and below the mesoderm you will be seeing the endoderm. So I'll just write it as ectoderm, mesoderm and endoderm. And this is nothing but, you can see the highlighted area, whatever I am highlighting, this is the primitive streak, and this is the primitive node. You can compare this one. This is the primitive streak which you are seeing in red color, and this is the one which is nothing but the primitive node. So what happens is, like, now I am using the red color to show the primitive node. So from the primitive node, few tissue will now slowly travel down into this mesoderm layer, and now they will travel. So as you know, where the primitive streak starts from, it starts from the caudal end, and it goes towards the cranial end. So you can see that, right? Caudal end and the cranial end. So this tissue which is coming from the primitive node, they will slowly enter the mesoderm and slowly they start pushing the mesodermal cells to the sides, and it will occupy the most of the region of this mesodermal layer. So now you can see that initially this is a tube-like structure, but later it gets solidified to form a structure called as the notochord. So now if you look at this picture, you can understand this is the primitive node which has induced the formation of the notochordal tube, and it gets solidified to form this solid structure called as a notochord. But this notochord has pushed the mesoderm layer, the mesodermal cells on either side. So you can see that on either side, this is the mesoderm, this is the mesoderm, this is the notochord, and above the notochord you are seeing the ectoderm. Now let us see how the neural tube is formed, because now already we are seeing this notochord. Now let us see how the neural tube will be formed. If you take a section of this plate, okay, how I will show is like there are few stages, 5 stages in the formation of this neural tube. So first of all, what I will show is like, I will show you this ectoderm here. Just underneath the ectoderm, you will be seeing this red color, that is the notochord. And on either side of the notochord, you are seeing the mesoderm. So I am just drawing the mesoderm. Okay, this is the basic structure what I have drawn from here. But this notochord, what will happen is like, it will induce the ectoderm, which is above it to form a thick layer. So initially this region of the ectoderm which was here, it was having cuboidal cells. Now as a result of this induction of this notochord to this neural ectodermal region, these cuboidal cells slowly turn into long columnar cells. So now this long columnar cell region is nothing but called as the neural plate, and on either side you can see other crest-like structure called as the neural crest. Okay, and of course this is nothing but what? The ectoderm, lateral to that. So what I need to say is like, I'll complete the diagram. This is the mesoderm and this is the notochord. So in this diagram you can see that this notochord has induced the ectodermal layer, a plate of the ectodermal layer to get thickened, where the cuboidal cells turned into columnar cells, and this part of the columnar cells is nothing but called as a neural plate, and lateral to it the neural crest is formed, and most lateral to it, it will be just like the ectoderm only. Okay, and this is a notochord. Just underneath the ectoderm you can see this is the ectodermal layer. Underneath the ectoderm you are seeing the notochord, and on either sides of the notochord you are seeing the mesoderm also, right? This part, this step is nothing but called as the neural plate formation. You can see the neural plate which is formed. From here what happens is like, this neural plate, whatever is there, okay, this neural plate will slowly start folding. So I am just now trying to fold this one. So this neural plate was started folding. So just below the neural plate you are seeing again the notochord, and on either side of the notochord you are seeing the mesoderm, right? So once this neural folding occurs, initially here a hinge is formed, which is nothing but called as the first hinge, and on either side you can see this is nothing but the neural crest, and just lateral to the neural crest you will be seeing the ectoderm. So if I have to label this, now see the neural plate which was initially flat, this neural plate, flat neural plate, now started getting folded. This is the neural plate, neural crest, this is the ectoderm, and this is the notochord. So after this folding, what happens is like another, I'll just write it as folding, right? Folding of the neural plate. And after this, what will happen is like this neural plate will start again, it will go inside, and the lateral sides, that is the neural crest region, will start getting elevated. So how it happens is like, I'll just show you. Now you can see that this is the neural crest region, okay, and this is nothing but the neural plate with the hinge, and below that you will be seeing the notochord again. Okay, so what is happening is here, the neural plate has went down and the neural crest has raised above. Okay, and just lateral to that you can see the ectoderm. So to complete the diagram, I am writing it as ectoderm. This is the neural crest, and you can see this is nothing but the neural plate. Okay, and this is the notochord. So this stage is nothing but called as the elevation. And after that, you within the same diagram you will also see next stage called as the convergence stage. In this convergence, nothing but you can see that this neural plate, whatever is there, I am just keeping dots for the neural crest. Sorry, this neural crest, whatever is there, it will slowly start coming into this groove-like structure, and this groove-like structure is now called as the neural groove. So the neural crest will slowly start coming into the neural groove, and the lateral part, whatever you are seeing, the ectoderm, this ectoderm will slowly start coming towards each other. As a result, what is happening is like the ectoderm are meeting with each other. Now the superficial most layer, topmost layer will be the ectoderm, and the neural crest cells have gone underneath the ectoderm, and now this neural plate has been closed. So finally what happens is like, if I have to extend this diagram, finally how it appears is like, now see the both the sides, the ectoderm has been united, right? This is the ectoderm which is united, and just beneath the ectoderm you are seeing the neural crest which was there. So this neural crest will be in the form of few cells only called as the neural crest cells, and underneath the neural crest cells this whole neural plate has joined to form a structure called as the neural tube. And just below the neural tube you are seeing another structure called as the notochord. This is the final stage, how a neural tube is formed, and just below the neural tube you will be seeing the notochord, and these are nothing but the neural crest cells. So this is all about the formation of the notochord. And coming to the functions and fate of the notochord. Basically most of the notochord degenerates, and few parts of this notochord will form the nucleus pulposus in between the intervertebral discs, and also it also will form the apical ligament of Benz. And most important function of the notochord is nothing but the induction of neural tube.